they've been adjusted.
Good morning, Dr. Tagmeyer. How are you, sir?
Thank you. I'm fine, Dr. Marion.
Dr. Meir, how are you?
You have chosen a
Hello.
wonderful and
All right.
look
Now
what they
that we
did.
have all the panelists on board and while people are signing in, we should go ahead and start for the sake of time. Let me share my screen with you. Let's see, share screen. Let me share this screen. You should be able to see my screen. I have too many of them on board. Let me reduce some of them so you can only see what you need to see, not the music, of course. Okay.
Is that a picture of Texas in the summertime now?
That's correct, in the background. So you see, do you see my screen?
Yep, there you go.
Good morning, everyone, and thank you very much for coming to our monthly webinar series, the Journal of Cardiovascular Aging seminar series. And I'll tell you briefly about the journal, and Dr. Yibin Wang will introduce our distinguished speaker, Dr. David Cass. I have Dr. Tagmeyer and Gabrielle Sciattarella as our panelists for today. So the journal we started a little less than 2 years ago is a peer-reviewed open access journal. And we are getting listed gradually on all major citation indices. We will be going to PubMed and PMC this month. And so far, we don't charge anyone for the publication fee because we're still in the developmental phase of the journal. We publish all kinds of articles, and we really define cardiovascular aging very, very broadly, no really specific, the typical geriatric issues. I mean, aging starts since the birth of a single cell. So you imagine the scope of it. We have a distinguished group of panelists on our editorial board and editors. And we're very, very proud of all of them for their contributions to the journal. We have, as of last week, we have started our third issue, third volume. The first issue, we have getting enough citations and getting enough tractions that we expect this journal to be ultimately make it as a prominent cardiovascular journal. We have overall about 50 articles published in PubMed, and we're getting interesting feedback about interest in our journals. This is our fourth webinar series by Dr. David Cass. We had 3 more before. Webinars are recorded, and after recording, some of them get quite enough interest and attention, including some have been apparently viewed by over 20,000 people. So that's this is, of course, after we post them online. So it's very attractive venue to discuss cardiovascular sciences. Dr. Yibin Wang, who was at UCLA, was the director of the cardiovascular theme program at UCLA for years, now is the professor and director of cardiometabolic syndrome in Duke National University of Singapore. Will make the introductory comment about our speaker, who is eminent physician scientist, and this speaker, by the way, this lecture has been named one of his, Dr. Cass's mentor, Keiji Sagawa, who was hemodynamic engineer, bioengineering expert in cardiac mechanics. So Yibin, you take over and introduce Dr. David Cass for the seminar.
Thank you so very much. I really appreciate and such a great honor and privilege for me to today introduce Dr. David Cass as the speaker of the Journal of Cardiovascular Aging webinar series. Professor Cass actually does not need any introduction to this audience. He is as defined, what described here, eminent physician scientist in our field. He obtained his bachelor degree from Harvard, followed by his MD training at the Yale Medical School of Medicine. I actually know him for many, many years, and who really had a tremendous impact on my career and on my science. And not only because it's the breadth of his knowledge in cardiovascular medicine and research, truly his generosity of sharing that, and also his incredible innovation in both medicine and science. So you can see that his expertise really expands from bioengineering to molecular biology to genetics. I'm sure he's going to tell you a bit more about omics, quite a bit actually today. And he's really standing at the cutting edge of our field. Really have no fear of bending the definitions or paradigms and identify really the novel insights of cardiovascular diseases. He received many honors, among them the Distinguished Scientist Award from American Heart Association and Louis Arthur Lucing Prize in Cardiovascular Disease, the Peter Harris Distinguished Scientist Award, and the Innovator Award from ISHR, and the Basic Science Achievement from American Heart Association, as well as the Outstanding Science Award from NIH. It'll take me many minutes actually to lay out all his awards, but I think we all want to hear his presentation today. It's no better person than Dr. David Katz to redefine HAPF from the ground up. Thank you so much, Dr. Katz.
Thank you very much for this really kind introduction, Eben. Marion, I think, I need to be disentangled from you, and then I can start my screen sharing as soon as you go.
You have to stop sharing, Marion.
Hasn't happened yet.
You are one panel shares. Let me, I guess maybe I need to,
You just have to stop sharing.
let me stop sharing. Maybe that's the reason.
There you go. There you go.
I want the culprit. Guilty. Oh, wow. All right.
All right. Everybody should now see just my slide.
Yes. Thank you.
Perfect. All right. Well, thanks very much again. It's a real pleasure to join this morning, at least for evening for many of you and elsewhere. And we'll just get started. So I guess I like to start simple. This is kind of a perspective of the cardiac cycle. You get about 1/3 of your time for systole and 2/3 of your time for diastole. And for the most part, the heart is able to manage life quite nicely. The issue of a disease, a heart failure syndrome, in which systole, if anything, seemed to be fine, which kind of meant diastole probably wasn't fine, was kind of simplified down to too much systole, not enough diastole by actually one of the folks at Hopkins back when I came here as a fellow way too many years ago. And it sounds sweet and simple. It's almost like a nice little jingle, but fundamentally it was kind of what we thought for decades. And the focus was definitely on diastolic dysfunction as the primary mechanism for why these patients who seem to have a ejection fraction in a normal range were still getting heart failure symptoms. I'm going to come back to the book of HFpEF myths multiple times in this talk, because I'm essentially trying to examine a lot of the things that we've thought And now look at kind of what's the recent data. Almost everything I'm going to show you, if not everything, is directly taken from human. The animal models in this syndrome are there. They're getting better. They still don't capture a lot of what actually goes on in the human beings. That's just, unfortunately, it's hard. You'll get an idea of why. So, one of the chronic ideas was that the problem is that these hearts are basically stiffer and diastole. They've got hypertrophy and fibrosis. And if we can concentrate on treating either of these two things, then we'll make them better. So what do we mean? A lot of people were focusing on relaxation, which is actually not what's shown on this slide. What's shown on this slide is a cartoon from ages ago, but you could find this in textbooks, where diastolic heart failure was defined principally by stiffening of the end diastolic pressure volume relationship, which is essentially diastolic compliance of the ventricle. And you compare this to systolic heart failure, where this is drawn more or less the same as it would be for a normal heart. It's the systolic process, or the ability of the heart to stiffen in systole, that's actually the most defective and depressed, whereas systole and diastolic heart failure is normal. So, almost binary, very simple-minded, but yeah, this was driving our thinking. And there's data. I mean, this is 9 years later. There's stuff even more recent than this from Mike Zile's group, where they had taken the epicardial tissue from patients who were actually going to the surgery for bypass surgery, but had, in sort of retrospective analysis, symptoms that would say they probably had heart failure with preserved ejection fraction. And those with both these symptoms and hypertension, compared to just hypertension alone, had stiffer myocardium. And it was shown in this study to be a combination of greater interstitial fibrosis and also problems of the large macromolecular spring inside the myocyte known as titin. We kind of looked at this even before that study, sort of halfway in between, just asking a simple question where we're going to compare patients with hypertension and LVH and actually other sort of comorbidities associated with HFpEF, but they're not going to have any symptoms. They have no heart failure symptoms. And we'll compare those patients to a group of patients who really have HFpEF symptomatology. And one simple question, this was all done non-invasively, was just how much diastolic dysfunction do they have? Is it really so different? Because it's supposed to be. Here's a control group that's age matched, but these patients don't have any of these comorbidities, compared to our HFpEF cohort. And these patients had all been admitted to Hopkins for shortness of breath, for real dyspnea and fluid overload, and they had to be managed. So that this is not kind of an outpatient, barely symptomatic group of people with, diagnosed with sort of exertional dyspnea. These are sick patients who really have heart failure. And sure enough, they have more diastolic abnormalities based on an echo criteria of Mayo Clinic Index, compared to completely non-comorbid and non-heart failure controls. But what about the group that have these comorbidities, but they don't have heart failure? Well, they actually look very much like HFpEF patients. And for me, over a decade and a half ago, this was kind of the beginning of the debunking of a myth that it isn't that they're just so much more diastolic dysfunction. And it has to take a lot more than that to actually produce the syndrome. Is it fibrosis? Well, not so fast. Although you can find fibrosis, and this is now work we published fairly recently from the Hopkins cohort of HFpEF patients, in which we're getting endomyocardial biopsies in these patients and looking directly at the tissue. So over 100 patients who had biopsies that went into this study. And in those that are non-amyloid, what we see is basically severe to moderate in only about a quarter. So close to 75% or 3/4 of the patients have mild fibrosis. It isn't all that different actually than what you would find in a donor control heart. With respect to myocyte hypertrophy, about 50%, it's quite mild. Only a very small number actually have severe myocyte hypertrophy, and about a little under 50% have moderate. Turns out we also found almost 18% of these patients had non-diagnosed amyloid. These are not patients who we thought had amyloid, who we then biopsied, but these are patients who we thought had HFpEF. They came in as HFpEF, and we didn't think had amyloid, and we found out they had amyloid. So it turns out, given that HFpEF is half of all heart failure, there's a lot more amyloid probably out there than we previously thought. If you look at the tissue and actually measure the fibrosis histologically, as opposed to the sort of qualitative scale, you can see the range that represents a donor control group, and there are a lot of non-amyloid HFpEF patients that fall right into that range. And this is not unique either for that matter to PEF versus patients with heart failure and a reduced ejection fraction. You get a lot of patients with similar levels of fibrosis. This is not a unique aspect of HFpEF, and a lot of them are really not all that bad. So jump ahead 20 years from this paradigm that we had throughout the 80s and 90s, that HFpEF really was diastolic heart failure, and that it was a fibrotic and hypertrophic and hypertensive form of heart disease in older patients, often women, to I think what is kind of the current view. And I use a camel, who's definitely has too much straw in its back. And all of these are the various components, the various straws that go into heart failure with preserved ejection fraction. We've expanded our concept of the comorbidities far beyond just heart stuff with LVH and then hypertension, to include prominently aspects of atrial dysfunction, renal disease, chronotropic incompetence, anemia, which is actually common in a fair number of patients. Not severe, but nonetheless there. Peripheral skeletal muscle dysfunction, and particularly on the metabolic side, and fuel uptake. And down here, obesity, diabetes, and metabolic syndrome. And as I would point out, not all straws are the same size. Some of them are really much bigger. And with pun intended, the really biggest straw these days, and this is becoming global, is obesity. US is one of the leaders in this, along with components of the Middle East. But these other countries around the world are getting darker too, as the prevalence of obesity has been rising. In the US, over 40% of the population has a BMI over 30, and that's kilograms per meter squared. A fifth of our population has what would be considered morbid obesity, exceeding 35. It's a major comorbidity for many, many medical diseases, not just heart failure or cardiovascular disease. We learned this up front and central when a brand new virus showed up a number of years ago that suddenly was taking over everywhere, and patients with severe obesity were doing considerably worse. These are now really the two most common HFpEF comorbidities. And the issue is that we've had decades to develop treatments for hypertension. We're just getting started with some of the new drugs on the market to begin to effectively treat obesity, mostly by reducing food intake with these drugs. So if you think about the camel, then you could step back and say, okay, so here's another idea. It's not really that the heart's that bad. It's just in a difficult situation. It's got this obese body, it's got dysfunction in the kidneys, the pulmonary, skeletal muscle, vascular, all these other systems are sort of colluding. And basically anybody's heart would struggle. I mean, it's like taking a car that's designed, for a small compact and sticking it into an 18-wheel tractor trailer truck. It's probably going to have a difficult time accelerating. And there's an alternative hypothesis to this, which is actually that the heart is actually messed up. And until fairly recently, I thought the first hypothesis was probably more likely. But I'm now more convinced that the heart is actually messed up. And why? Where did this come from? Well, again, we've been taking these endomyocardial biopsies in HFpEF patients. And these aren't from the left side. We don't go into the left ventricle. We are taking them from the right ventricular sort of mid-septum. But in work that we published, and I'm not going to show, but I'll show other data from the same paper, we've sort of looked at when we have the whole heart, where we've done comparisons, and at least this side of the septum and this side of the septum look pretty much the same. I don't know about the LV free wall in all cases, although from a transcriptomics level and other forms of heart disease, that they all look pretty similar. So this was a study we published just a few years ago, where we took about 120 patients in which we had biobanked endomyocardial biopsies, and we divided them into 3 groups. And we had a group of patients where they were in the upper quintile of levels of systolic blood pressure and left ventricular hypertrophy, but in the lowest quintile with respect to obesity and diabetes. And we gave these simple, what quintile you're in, you got sort of a score, 0, 1, 2, 3, 4, and we create a ratio. And so they're going to have the highest ratio. And then we have the opposite group, the blue group. They're going to have the most obesity and the most diabetes, but the least amount of hypertension and hypertrophy. So they're down here. And then we got a group of patients. Each of these are roughly 15 to 20 patients that are right in the middle. They're going to have just as much hypertension and hypertrophy as the red group, and they're going to have just as much obesity and diabetes as the blue group. So by average, when you create the ratio, they should fall in the middle. And the BMIs follow the way we described it, and their LV mass index is as we described how we created these groups, as are their systolic blood pressures. And importantly, they all have basically the same, not normal elevated, but similar pulmonary artery systolic pressures, pulmonary artery capillary wedge pressures, ejection fractions. Very, very few of them are anywhere near 50. Our patients are basically mostly over 60% ejection fraction. And perhaps not surprising, but wasn't really into any of the calculations of coming up with the groups. Their log NT-proBNP levels, as a log scale, is about an order of magnitude greater in the group that has mostly hemodynamic load type phenotype and the least amount of obesity. So what is their sarcomere doing? That was basically our question. We isolated myocytes from these biopsies. We chemically treated them so that the membrane becomes permeable to solutes. All the stuff on the inside, with the exception of sarcomeric protein, basically leaves the cell. And we can now expose them to different steady state calcium concentrations. We've got a force transducer and a length controller. We fix the length at approximately 2.1 microns, and we look at different tensions that develop, which is force divided by cross-sectional area, as a function of steady state calcium. And here's a typical sigmoidal shaped tension calcium relationship in a control group. These are non-failing donor hearts that weren't used for transplant. And this would be sort of the range in diastole, where the calcium levels are low. And then this is sort of normal systole in this range. And as I'm going to argue, people have often in the muscle field have sort of viewed all of this stuff up here as non-physiologic. I disagree, and actually think a lot more that's going on up here is relevant to exertional symptoms. Not necessarily rest, but definitely exertional symptoms. And this range is actually quite important. So this is what happens in a patient with heart failure, a low ejection fraction, right ventricular failure as well, who's about to get their heart transplanted. This is the explanted heart from this patient. And we've known what this looks like for a long time because we've had access to this tissue for a long time. And you can see in systole, in sort of the systolic range, not so much the diastolic range, there's already pretty profound depression of tension development at similar levels of systolic calcium. Way out here is non-physiologic, but in this range, I think this is still quite relevant. Okay, what about the blue group? They were the people in HFpEF, the red group, who they have hypertension and hypertrophy, but not so much obesity. Well, in systole, they look pretty much like controls. And this is very concordant with data. The only other data really that's out there that goes back almost 20 years, work that Walter Paulus and his colleagues had published in biopsies that they were getting from the left ventricle, actually, in what they call diastolic heart failure patients. But these patients typically were very hypertensive and hypertrophy and had BMIs at or below 30. Our BMI median in this group is about 40 plus. In the diastolic range is a little bit of sensitization, which could contribute to diastolic tone. But what's important is that in systole, things look basically okay. Well, what about the patients who are mostly obese? they look just as bad as HFrEF patients. It's profound depression of systolic function. Diastolic properties are actually not as stiff as the ones without as much obesity. And what about the green group? The green group, remember, is a mixture. They have just as much hypertension as the red group, and they have just as much obesity and diabetes as the blue group. So which one wins out? And it turns out it's obesity that's a real driver. It's not diabetes. You can have just as much, or you can have diabetes or not have diabetes, or at least manifest with hemoglobin A1C. I think that's shown in this. Here we've separated them with colors, but whether you did have diabetes or did not have a diagnosis of diabetes. But if your BMI is high, the tension that you can develop at systolic levels of calcium goes down. And if your BMI is lower, it's higher, with the normal range being up in here. And this being almost as bad, down at around 10, is really as bad as a HFrEF patient getting a transplant. So how is this possible? These patients all have ejection fractions of 60 or more. How could you have such profoundly abnormal calcium dependence of tension development in your sarcomere? Well, for one thing, ejection fraction is not contractility. We teach this to medical students. We teach it to all of our clinical cardiologists. Just because we've focused on this, we've almost perseverated on ejection fraction to the point of defining heart failure by different levels of ejection fraction. It's very convenient, but it really doesn't tell us very much in terms of the biology. That's for sure. But more than that, I think it isn't even that EF may be preserved, and it's preserved at rest in a patient sitting in a chair, but not so much when they actually get up and do stuff. And this is really a functional reserve disease. We define heart failure often not by just what the patient's doing at rest, but their inability to actually do even moderate exertion. So this is work from my own group that's getting back almost 30 years. This is a frightening thought. But here we plotted the end-systolic elastance, which is a chamber level stiffness, similar to the stiffness that the myocyte can generate at end systole, for a control group. And this control group was not hypertensive, or they really were a pretty healthy group of patients. It's all human. And then this is a group of patients, we called it at the time hypertensive hypertrophic heart disease, but all of these patients had heart failure symptoms. That's what brought them to the cath lab. And these days we would call this really HFpEF. And what we're showing here, I keep drawing, I'm pointing to the wrong ones, but what we're showing here is that the ones with HFpEF down here have very little change in contractility, essentially nothing, as we increase their heart rate. We're using heart rate as a stimulation. Normally, contractility is supposed to go up, and in fact, in the control group, contractility does go up. It almost doubles as you go from a heart rate in the 70 range to a little over 150 beats a minute. It's a classic positive force frequency response, but it was very blunted in these HFpEF patients as a reserve mechanism. And Barry Borlaug, a number of years later, about 17 years later to be precise, also demonstrated in the cath lab that HFpEF patients' ability to increase contractility, measured several different ways, all with pressure volume relationship analysis, is depressed. This is shown in red, as compared to a group of patients like we had, where they had hypertension and LVH, but they didn't have HFpEF. They didn't have heart failure symptoms. They're in green, and they're similar to the control group. So PEF, preserved ejection fraction, may exist, but that doesn't mean there's preserved contractile reserve. And I think that's what our sarcomere data are really pointing to. Further evidence that this is relevant to exertional symptoms comes from a prior study of ours, where we were looking at patients with RV failure who either had systemic sclerosis, pulmonary hypertension, or idiopathic pulmonary hypertension. When we looked at their myocytes, interestingly, we saw, compared to the control group, very similar to ones I've been showing you, the systemic sclerosis patients have similar kinds of depression in the systolic range of calciums, not so much in diastole. And whereas the other patients with pulmonary artery hypertension, but not due to systemic sclerosis, due to a cause that Maybe some genetic cause could be related to some of these BMP proteins, whatever. They're actually higher than normal. What was interesting about this data set is that it's the first time it's ever been done where we could compare this to what does their contractility do with exercise. Now we're not doing pacing, but we had them in the cath lab supine with a bike exerciser. And indeed, the patients whose sarcomeres look like this, which is very much like what our obese HFpEF patients look like, have very little change in contractility, essentially nothing, during exercise from stage 0 to 2. Doesn't take a lot of exercise. They pretty much poop out at this point. Whereas the group where this was enhanced show a increase in contractility of the whole heart. Same patients. These are the same patients from which we got their biopsies with exercise. So I do believe that rather than just dismissing what goes on up here as non-physiologic, because these are high calcium levels, I think it's really the thing that's correlating the most to actually exercise reserve contractility. And in studies that are ongoing, we're now in the HFpEF population, we're now looking at structural abnormalities in the sarcomere, trying to understand why obesity is doing what it's doing. We've been looking at whether we can reverse this by changing post-translational modifications. I'm not showing this right now because this is still unpublished data, and this is going to be publicly posted, but it's coming. And we're looking at various ways of activating, really enhancing sarcomere function, something that we didn't think we needed to do in a patient whose disease was called preserved ejection fraction. But I think with the growing population of severely obese patients, this is really what we need to do. And we should be able to target it to the right ones, really kind of looking at their body mass index. It may not actually take that much more personalization than that to get started in targeting the right people. I thought I'd give you just a hint of what the structural things are that we are now doing. This is a paper that's in press. It just was accepted recently. It'll be out shortly in circulation, where we're looking with X-ray diffraction patterns in human biopsy material to visualize the distance between the myosins, which is what's known as lattice spacing, as well as the interaction distance between the myosin, the larger circles, and actin myonofilaments, the smaller purple circles. And this ratio of this distance to this distance is classically used as a marker of engagement of the thick and thin filaments. Just a blow up where you can see these diffraction intensities in the meridian to determine these differences. What's shown here is what happens in a non-failing control. These are going to be patients with HFrEF, not HFpEF, but HFpEF's coming. And at high calcium, you can see this, this I11 to I10 ratio basically goes up. So we're getting more engagement of the actin and the thin filaments with myosin. And in a group of patients who have compensated right ventricles and HFrEF, this also goes up significantly. But when you have HFrEF that's decompensated, and by decompensated, depressed forced calcium dependence, it's very, very, very similar kind of behavior to what I showed you for HFpEF in this group. What's striking is that now, even by increasing calcium from, this would be 10 to the -8, so 0.01 micromolar, really low diastole, to 10 micromolar, very, sort of really super activation state. There's very little change in these distances. There's a structural impediment to contraction that's taken place when we're seeing this. So we're trying to tease out that role in HFpEF now. With all that heterogeneity on the camel, another myth would be that there's no way to find consistent molecular signatures because everything's going to be too complicated and there are too many comorbidities. And I think that turns out to be false. And that was based on a study also published just about 2 years ago, where we did transcriptomics in patients with HFpEF and HFrEF and our donor controls. And with principal and component analysis, you can see these things really do separate pretty clearly. So at least at the transcriptomic level, this HFpEF signature is different from HFrEF and more cohesive, despite all the different mishmash of comorbidities. And if you adjust this for sex, age, BMI, glomerular filtration rate, and diabetes, you still basically get separation. The vectors all shift around. That's what happens with principal component analysis when you introduce other terms, other vectors, but they're still basically separated. So that begins to take away from the idea that if this is going to be hopeless to ever kind of target a molecular signature, a particular molecular target, because it's, you know, everybody's going to have too much of different things and it's all going to be complicated. If you look sort of a little more granularly at what's going on with genes that are upregulated, a lot of the upregulated genes turn out to be in the respiratory chain, electron transport and oxidative phosphorylation. Interestingly, as we're about to report, at the protein level, things are quite different than this. This may indeed be more of a genetic nuclear response to what's going on that's actually depressed at the protein level. But nonetheless, what was important here is that this seems to be very correlated with BMI. So being obese is one of the contributors to whether these things are going to be up or down in a HFpEF patients. This is purely in HFpEF. When we adjust for all the comorbidities, then basically we can explain the variance in the transcriptome for these complexes entirely. So none of them remain statistically significant by themselves compared to unadjusted. What about the downregulated genes? The downregulated genes are not impacted at all by either BMI or any of these comorbidities. And interestingly, the downregulated genes have a lot to do with protein trafficking and processing, from Golgi vesicular transport, autophagy, endomembrane system organization, vesicular organization, localization of nucleus. I mean, it's a very interesting and cohesive set of genes. And we and others are currently really digging deeper into what this is telling us that's abnormal, and the kinds of processes that we need to start to target therapeutically. This has not been the usual suspects. These are different suspects. We can also identify subgroups purely from the transcriptome. And that's shown here, where we use non-negative matrix factorization. We fed in about 3,000 of the most differentially expressed genes into the algorithm. It's a machine learning tool, and it spit out these three groups. These are the two particularly most coherent. This one was a bit more mixed. And they turn out to have different clinical features. Again, this is purely based on the transcriptome. But clinical feature number one, which is a big one, probability of event-free survival, was much worse in the group that had this particular transcriptomic signature compared to this transcriptomic signature, which is in group 2 up here. The mixed group, the third group being this one. But this is statistically significantly different. So there's something going on here in these genes that is predicting worse survival in HFpEF patients. So what are the things that might be going on? And the specifics of the genes, I'll send you back to the paper, which has been published. But just to point out that indeed group 1 has kind of the most evidence of hemodynamic load. These were the most hypertrophied hearts. They had the highest pulmonary systolic pressures. They were not any more dilated than group 3, but they definitely had the highest NT-proBNP as a marker of hemodynamic stress, and really much higher in median compared to the other two groups. And this was very statistically different. The other interesting thing is that the group with the lowest mortality was 100% female, purely predicted by the transcriptomics. So this is work in progress to further try to figure out whether we can come up with identifying specific subgroups really based on molecular signatures, not just clinomics, not what's going on necessarily in their blood. We've been looking at that. It generally is not as predictive as what's going on in their hearts, at least in defining subgroups. And I'll show this next in my last myth. So because these patients, or many of them, have marked obesity and type 2 diabetes, we would anticipate that they can't really metabolize glucose. They've got, at the minimum, insulin desensitization. They may have frank evidence of high levels of circulating glucose, Many of them were on anti-diabetic drugs. It's all type 2 diabetes. But because they have a tremendous amount of fat, it has been generally thought, and mostly based on animal models of both diabetes and obesity, that they're going to take up the fat and effort to burn the fat. They may not be able to do it so well. Hearts often show in those kind of animal models lipotoxic manifestations with an accumulation of fat inside muscle cells that isn't really being effectively burned, but still a fair amount of lipid oxidation has been thought to take place. So when we summarize what the healthy heart can do, the healthy heart is really able to eat almost anything. It prefers fatty acids. That's the majority fuel. There are some papers out there that are suggesting remarkably little glucose is consumed. So sort of surprisingly remarkably little glucose. They also have the ability to shift if there's a fuel limitations. And in particular, the hearts can catabolize ketone bodies and lactate actually quite nicely, if available, as well as a number of amino acids, the branched chain amino acids, for example. So they're omnivores. In HFrEF, it's long been demonstrated that the ability of the failing heart to utilize fatty acids goes down. Ketone body utilization generally goes up. Lactate goes up to some extent as well. There's more, at least glycolysis present. Whether glucose oxidation is limited or not remains, I think, a little bit more controversial still. That's my impression from what I read. We've not directly looked at this ourselves, but fundamentally, I think the main take home has been that fatty acid oxidation is compromised. And as I said, it was kind of speculative what was going to be going on in HFpEF. We just didn't have the data because we didn't have the hearts, we didn't have the myocardium. We don't transplant HFpEF patients, so we don't get the material generally. But as you now know, we get biopsies. So this paper has just been now officially published. It was online just a little earlier this year. And a couple of interesting things. We did essentially a somewhat limited metabolomic survey concentrating on acyl carnitines, which are the fats that have been brought into the mitochondria with the carnitine shuttle. We also looked at organic acids, like the Krebs cycle intermediates, and all of the amino acids. And when we just look at the metabolome, and I'm not showing you the heat map that goes with this, principal component analysis, once again, seems to be able to separate what's going on in HFrEF from a control and from HFpEF. So they have a unique metabolomic signature. This is not just going to be, more HFrEF, and it's certainly not normal. This is what you'd see if you looked at blood plasma instead of heart tissue. And in the plasma, there is no particular unique signature. It really overlaps a lot. And this is previously, before our heart data, this is really all that was out there, was efforts to look at the metabolome in plasma. There are things in plasma that are interesting, but I feel pretty confident they're not reflecting what's going on in the heart very well at all, if at all. And I think that may be true of a number of other kind of biomarkers that people have been looking at in plasma. They probably are coming from other sources, lung, vasculature, liver, kidney, I don't know. But the heart is not a major source, I don't believe. One of the interesting real surprises in this study is that fatty acid oxidation, instead of being up, which is what has been described in HFrEF, is actually reduced. And the signature at the transcriptomic level is shown here, highlights this already because you've got genes for PPAR alpha, PPAR gamma, phosphoprotein lipase, lipoprotein lipase, sorry, tongue twister, CPT1A, and CD36. These are fatty acid uptake, key uptake proteins, both into the cell and then into the mitochondria. These things are all down-regulated, and we're seeing similar sorts of changes at protein levels also. If you look at the metabolites, the acyl carnitines, and this is a flower plot where the vector, the farther away you are from the origin, the higher the magnitude of the particular species, going from 8 carbon all the way over to 22 carbon around this way. Well, this is control. The HEF-REF, which is shown in the pink color, is very reduced. And this was, again, long known. And a signature of reduced fatty acid oxidation, something that we thought was certainly compatible with that. Well, it turns out HEF-PEF looks much more like HEF-REF. It's certainly not up. It's not increased. Again, this is myocardium. If you look at what's going on in the blood, it's completely different. And if you had just done your metabolomics analysis from the peripheral plasma, you wouldn't have recognized that there's something very wrong and very limiting about what's going on with fat metabolism in the heart muscle itself. Lastly, branched chain amino acids also turn out to be strikingly different. They're highest in the HEF group in blue. Leucine, valine, isoleucine are the 3 branched chain amino acids. And their catabolites, which are over on this side of the plot, are the most reduced compared to the controls, which are shown in green. This is interesting and potentially important. These 3 species are known, among other things, to be a potential fuel for the heart, but the fact is that these are down suggests they're probably not really getting oxidized. It doesn't look like they're being used as such. And their levels are higher, I think, presumably because of a bottleneck of catabolism, but importantly, they can also then stimulate enzymes such as the mTOR complex, which can be pro-hypertrophic and actually favors glucose over fat, tends to be lipogenic, and engages in a number of behaviors that in a failing heart are probably not all that useful. They're, if anything, a bit more maladaptive. And this just sort of summarizes all from this paper, what's going on. These downstream species are downregulated, suggesting a sort of a catabolic block, whereas the upstream branched chain amino acid species are the highest. So are we getting any closer? Well, it's interesting that the only FDA-approved evidence-based drugs that are on the market now for HEF-PEF are basically diabetes drugs, sodium glucose transporter inhibitors, the SGLT2 inhibitors. We still don't really know exactly what they're doing in the heart. This doesn't even seem to be expressed in myocytes, and there have been knockout models of this in the heart muscle where you still get what appears to be a benefit of these drugs. So, you know, work in progress, but nonetheless, it was somewhat ironic for a disease that is named after what its ejection fraction is, and the symptoms ends up currently being treated very effectively, really. The first kind of Kaplan-Meier type stuff we've ever seen that looks like this with drugs that are otherwise for diabetes. Is there a way that we might be able to improve things by reducing just the obesity? That's now being tested with the GLP-1 agonists, a couple of ongoing trials, I think, that are going to give us a bit of a clue. I'm optimistic because I think this is really a big problem for the field. Our own contribution to this, and whether, because those drugs typically reduce appetite, we're trying to see whether we can burn fat. And we went to a nitric oxide natriuretic peptide signaling system related to cyclic GMP, which then activates protein kinase G. It's this side of the equation that has been previously linked to fat lipolysis. And we now know the phosphodiesterase that regulates this side of the pathway, as opposed to PD5, which regulates the nitric oxide side of the pathway. And we have inhibitors for PDE9, and we studied one of those and published this in late 2021. And we can take very, very obese mice on a high-fat diet with some pressure overload, this happens to be females with ovarectomy, and treat them with a PDE9 inhibitor, and we get a reduction in weight. So body weight is going up in the placebo arm, but is actually going down in many of the mice that were treated with the PDE9 inhibitor. And here's their total body fat mass versus lean mass. And importantly, this is not because of food intake change. This is not an appetite suppressant, unlike the drugs we're using currently in humans. Nor did it really change total activity in most of them. Some mice went more. And further, we see reduction in fat accumulation in the liver with a PDE9 inhibitor, and the rebrowning of brown fat, where instead of getting big hypertrophied lipocytes, you get more normal looking adipocytes in brown fat with increased mitochondria. And as a member, I showed you in HFpEF patients, things like PPAR alpha, genes associated with fatty acid uptake were downregulated. A PDE9 inhibitor seems to be upregulating these same genes. So there are now clinical trials that are testing this in both HFpEF and HFrEF. We'll have a readout probably by either the end of this year, I believe 2024. So I'll end with this slide. I think it's time for sort of a paradigm change. It's clearly far more than fibrosis and diastolic dysfunction, hypertension, and hypertrophy. The syndrome is now dominated by obesity and metabolic syndrome, which are colluding really to change myocyte systolic reserve, surprisingly at the sarcomere level, as I've shown you. I think new treatments that are targeting these metabolic features are really sorely needed. And I think the SGLT2 inhibitors are really just tickling the surface. There's a lot more that needs to be done. And with that, I will stop, take questions. I want to thank the many people that are involved. This is a good list for the papers I've shown you, but the new stuff we're doing, I'm going to have to add more pictures. So thanks very much. I'm going to click out of this and I'll go back to stop sharing.
This is amazing. Wow.
Happy
Thank
to take
you.
questions. Sure.
Great.
My
Great.
pleasure.
It gave me really goosebumps remembering my early days as setting up my own lab. I learned PV loops from you and see how that.
They're still useful.
Yes, and I still see that how you focus on the clinical questions and provide so much more insight beyond the mechanism, but also therapy. And of course, you know, the branch amino acid is something very close to us as well. And seeing that it's actually being implicated, I remember in our lab meetings, we were very excited to see this. So maybe we can open up to questions to our panelists and also to our audience. And I can see Gabriel is actually opening up, and so ready to go, Gabriel?
Yeah. Well, thank you so much, David. This was outstanding as usual, really, from clinical to the basic mechanism and backwards. So also, particularly in the spirit of the journal, you know, the seminar hosting now, which is a Journal of Cardiovascular Aging. So can you elaborate a little bit more about what is, in your opinion, the role of aging in HFpEF pathophysiology? Because, you know, it's known that, I mean, it's been associated that HFpEF is more prevalent in older subjects compared to milder, although as you might refer, this is perhaps not the modern HFpEF that we are seeing now with the increase in obesity, and we know that your cohort of Hopkins is fairly younger compared to the others. So what do you think will be the aging component? So where the old HFpEF patients are in the calcium tension curve that you have shown now?
Well, so we've looked, each time we do these kinds of analyses, whether it's omics or it's like cellular physiology, to the extent that we can, we look and see, is there an age effect? I mean, in order to do that, you really do have to have the larger numbers. So for the cell studies, we're very happy to get data in, let's say, 15 to 20 patients. That's too small a group to make any kind of meaningful subgroups. I think the main comment that you've made, though, already is the one I would want people to think about. Yes, when I started my career and when I first got interested in this in the late 80s and 90s, doing pressure volume loop studies in humans and realizing that what we were thinking couldn't be right because they didn't have what was in my textbooks. But I was looking at it and I knew something was wrong. I didn't know what, and I didn't know how long it would take me to figure anything out, but I knew something was wrong. They were older. They were typically older. But obesity is affecting kids. It's affecting teenagers everywhere. And the planet is not only getting heavier because of people, but the age at which this is taking place is happening earlier and earlier. I also think that it used to be much older women, and more predominantly female. The sex disparity is also declining. I think we're seeing more and more getting close to 50-50. And part of that may indeed be men even earlier tend to put fat in their stomach, and it's a visceral fat. They're the ones who are probably more likely early, premenopausal women put it more subcutaneously. It's different properties, they're less pro-inflammatory kinds of fat. So in that sense, men now become more at risk actually for developing this, both earlier because obesity has become the dominant comorbidity, and because they're men, because they've lacked the estrogen component, which I think helps sort of fat catabolism and distribution. Estrogen itself is kind of PPAR alpha-like effects. So in that sense, I think it's all changing. No offense to the journal and the idea that it's interested in aging. I'm not saying age doesn't help you. I suspect, the older you get, the more you accumulate everything else too. But it's just, it's changing a lot.
Great, thank you. Dr. Takmir?
Is it me? Heinrich,
Yes.
or-
Yes.
Yeah, wonderful.
Yeah,
Thank you, David, very much. That was a whirlwind and wonderful. It reflects really the complexity of metabolism. And I think all of us spectators have their special actors. And
right.
You've been focusing on a number of actors, and other people focusing on a different set of actors. I'd be interested, David, in your opinion, but two things. First of all, the concept of metabolic reserve of the heart. And that is a phenomenon that applies to the normal heart and a diseased heart, and what are the main features of the metabolic, just as there is a contractile reserve, what fuels and supports the metabolic reserve. And the second question is, what do you think of my pet hypothesis, unloading the heart from especially the HFpEF heart from metabolic stress? And we're all familiar with hemodynamic stress, we're all familiar with neurohumoral stress, and pay relatively little attention to the metabolic stress, which can be modified, as we're witnessing, day in and day out, and bariatric surgery, but also, the SGLT2 inhibitors, and who knows, even metformin and unique properties of metformin, linking substrate oxidation to a greater intent than other hormones to substrate uptake. And then Gerald Reben's hypothesis, Lou Landsberg, and Jim Young of the catecholamines in obesity. So I'm stopping now and
well, you've laid out, I think, the future of what my lab and others is now going to be trying to address for the next, not quite decade, or we'll see how long ago. But I agree with you 100%. I mean, one of the things that kind of stunned me about our metabolomic signature, which is it's far from complete, and we're now filling in a bunch of other pieces to get a fuller picture, is just how fuel inflexible the HFpEF heart would seem to be. If we thought the HFrEF heart was fuel inflexible and lost some of its ability to utilize anaplerotic sources to compensate for other problems, add diabetes, add the inability to basically get sugar in, and simultaneously take the heart failure part of lipid catabolic processes and depress those, not use branched chain amino acids either, you're left with probably lactate And actually, I think amino acids, the one thing that seemed to be up the most in our heart muscle biopsies were a bunch of amino acids. And I haven't, again, this is all in the paper, and I have time to go through all of the other components, but there's a story there. And we're beginning to look at a lot of other components of what you refer to as the of metabolic stress. Exactly, I like that term. What are the transcription? What specifically do you need for appropriate transcription of oxidative phosphorylation genes and an enhancement of the ability to generate ATP? Because they don't look normal actually in HFpEF. I didn't go into that either, but some of that's in the transcriptomic paper. You just have to dig through. But we're finding this, and why is it abnormal? And what would control, both transcription and translation in particular? We're looking at that. There are some other interesting things that are going on the acetylation side, and because of abnormalities of Krebs cycle intermediates that we report. So we're looking at what that might be doing. Obviously, that has nuclear implications, which then turn into transcriptional signatures that are contributing to this sort of metabolic stress. And fixing that is, I think, exactly what needs to happen. The only things I think we tried, more in HFrEF than PEF, if I recall, is we tried to block fatty acid oxidation. I think we used a drug that, and when looking at our own data, I wouldn't have guessed that that would be the best thing to do, actually. I mean, I don't know what you feel, but to me, it looks like the heart's doing it already. And I don't know why that's necessarily going to help. And it's not as if blocking that is somehow magically going to fix diabetes and fix insulin, desensitization.
Yeah.
So all you're doing is adding kind of a little bit of a fuel and flexibility drug on top of a pre-existing fuel and flexibility. We need to be cleverer than that, I think. The good news is we haven't tried a lot of the things I'm thinking of now. And I'm sure you thought of these all too.
David, I could not agree with you more. And since I am probably the oldest one on this panel, I'll tell you, years ago, people thought metabolism, everything is known and nothing further needs to be known. The opposite is true. The horizon is infinite, really.
I agree. My comment to the world is that, and you'll like this, is that as a math-oriented hemodynamics person, I looked at some of these metabolic charts, which look way worse than any Intel computer chip diagram. And I just got scared away from it. And it's taken me 40 years. And now suddenly I'm have to do it because it's important, and it's obviously important. And as you say, we know actually remarkably little. Maybe the Intel chip analogy in that sense is true. It's quite complicated, but it's incredibly important to get right.
Yes. So Dr. Cass, I have to move on then, because there's quite a few questions from the audience, from the chat. How about I read very quickly and then you answer in a very short translations. Okay. So the first is about the hormone hormone replacement therapy. Do you see that in a woman subclass, when they have better survival, are they actually correlated with the hormones replacement therapy status?
There were only, so the short answer is I don't actually absolutely know the answer to that, although we would know if they were on it or not. Most of these people, they're all postmenopausal, that's for sure. There were only, I think, 10 in that subgroup, and it would be hard to imagine we're statistically powered to, even if we knew what it was, no matter what the distribution was, to really figure out whether that is helpful or not. I've been certainly I'm sensitized to the fact that estrogen, whether given as a hormone replacement or your body before you're menopausal, definitely has many important manifestations that could interact the things that we're looking at. So it is potentially relevant. I don't know that would explain, though, the NT-proBNP median being over 1,500 in the one group with the highest mortality, and literally 50 or 40 something, in that subgroup. I don't think that's going to be just, estrogen replacement.
It's actually quite difficult to generate this causal relationship. It's correlation right now.
Yeah.
So the second question is actually the sarcomere data that you clearly see differences between the HPF. From the obese conditions, actually you see quite similar patterns of sarcomere function between the HPF and the HFrEF, and yet the HFrEF targeted therapy to improve sarcomere structure or organ function actually failed in the HPF trials. Why?
You just put some words in there.
I saw that's
the
what
therapy he's talking about are just anti-HFrEF drugs, and the point there would be we actually don't have a successful sarcomere enhancing HFrEF. The FDA just declined approval of omecamtiv, which was the closest we've ever gotten. Yeah. And the drugs that we have been using have been all neurohormone blockers, like angiotensin receptor blockers and ACE inhibitors. And that's really not the primary problem with this syndrome. That just isn't. And we've, if anything, what I think is remarkable, and hopefully our data at least makes people think twice, is that for decades we used pretty potent calcium channel blockers and beta blockers in these patients, and they are not known to increase contractility. In fact, I can guarantee that the doses we were giving, they were decreasing it, and we were often using it because they had hypertension and hypertrophy. Now that they're more obese, I would question those drugs very strongly. So I think we just have to rethink things.
No, I agree. Actually, I see there's an effect of gastric bypass.
It's been looked at in small trials. We have yet to get a very large trial together. It's complicated because the surgeons generally don't like to see, a patient they're taking to the OR for something like that with a diagnosis of being hospitalized for heart failure, several times. And so it's been avoided. Now that we have almost gastric bypass-like effects from the new drugs going at appetite, I think that's where we're going to learn this on a large enough sample size to figure it out.
That question from Marinja is the status of inflammation in HPF. I'm also sharing that question from your transcriptome studies. So you don't seem to actually identify inflammation as one of your major, at least from the PCA components, the key pathways affecting the HPF.
I think it's there. I will say that the question of how much of it is really in the heart, and remember, most of the transcriptomic data I showed you is myocardium. I think it's not that much. I mean, we don't see it in the biopsies of these patients. We that it's all been evaluated by our transplant pathologist. It certainly doesn't look like typical myocarditis or much inflammation. And so, but that said, I think it's very important. I think macrophages are really important. We are looking at these things. I just don't think that it's going to be all in the heart. I see from Peter. I can see the questions in front of me too. You can. Great. What could be the driver for systolic dysfunction in HFpEF? That's the bazillion-dollar question, Peter. I can tell you that we are looking at this structurally. I showed some X-ray diffraction data for REF. It'll be coming, I think, hopefully by the fall, but we've already got some preliminary data. I can just say that there definitely is something going on structurally that appears only in the obese subgroup, which is quite interesting. So, and we've got EM that actually shows some very striking differences, whether you're very obese or not. So structurally, I think it's good. There's going to be something going on. And then we've been looking at PTMs, and we've got some phosphorylation data. We just don't have it pinpointed yet as to exactly what was dephosphorylated or phosphorylated. That's work in progress. We're struggling with our proteomics in such tiny pieces. It's been hard to get good phosphoproteomics from 3 milligram pieces. There are also HFpEF patients who have small size frail old ladies without much obesity. What do we think about? I think that's kind of the old HFpEF, right? And we used to be, we called them at Hopkins LOL before that was laugh out loud. It was little old lady. It was little old lady hearts who had, small, and I get in Asia, in parts of Asia, this is still a more common phenotype, although obesity is increasing there too. And it has probably a somewhat more hemodynamic oriented component. But that being said, I didn't mention this, but the metabolic signature that we've published and I showed actually doesn't segregate by BMI. I thought it would. I was stunned that it didn't. Helmut probably would say, yeah, of course, that's what I expected, but it did not. And so in that sense, we tried things that should have worked in the little old ladies. You know, we can treat blood pressure. We have drugs to treat hypertension and hypertrophy, and they weren't getting better. So I think metabolically, it's probably also different. And I think we kind of missed it, but it is a different phenotype. Maybe the last question, do you think glucose oxidation beneficial during cardio? Yeah, I mean, this gets to I think what Helmut was saying, that, I mean, that's one of the classic reserve mechanisms, that you tend to switch over and use more glucose when you have to do something quickly, and where your ATP, your oxygen requirements are definitely less. You don't get as much ATP, but your oxygen requirements are less. I just don't know how that's going to work with someone with a BMI of 50 and, you know, striking insulin desensitization. It's not, I think that's where the weight loss therapeutics may turn out to be absolutely critical to get past some of that bottleneck, so that fuel reserve with glucose can be used again.
Maybe David, you can give a last shot at Lori's question.
Yeah.
I want the answer too.
Tail name, high-fat diet, mouse models fit with human data.
That's from Gabriel.
Right. Well, with all due respect to Gab, sometimes they do and sometimes they don't. So with respect to the sarcomere, they don't, at least so far. We're tweaking it a little bit. We have an idea we're going to play around with to see if we can figure that out. There's, with respect to something I haven't talked about or shown any data on at all, but turns out we've been looking at histone, PTMs, and sort of associated transcriptomics. His model looks amazingly good compared to human, and much better, for example, than the ZSF1 rat, which is another popular model, or one of the pig models. On the other hand, we're also looking at a lot of different metabolic pathways, specific metabolic pathways. And if I brought the postdoc in who's working on that, and he would answer your question right now, he'd say, yeah, not so much. There are things that it definitely does. I think their ER stress finding and the signaling was very much a part of human HFpEF. That was a big focus of the original paper. And that also gets to abnormalities of processing, protein processing and quality control. I think those are real. But yeah, it's complicated, and it's not really that surprising. I mean, human beings, I don't know any disease of human beings that basically is a mouse on LNAT. It did a bunch of things that are interesting. It was, you know, it's tractable for sure, more than some of the other things people have been doing. It's being used all over the place. It's probably the most prominent model ever. I'm a little concerned that it's being used a little bit too glibly, I suppose. I would like to see more people look at our transcriptome now that it's published, and look at our metabolome now that it's the proteome's coming in human, and then start to ask whether the things they're interested in looking at are in that model or not. It's not just all about EE prime and hypertrophy and, you know, do they have something that looks like, and they have a normal ejection fraction. We've been phenotyping our animals kind of the way we were phenotyping humans 30 years ago. And we need to start using kind of the signatures that are now becoming more cellular molecular as they're being developed from human to better pick the model and sort of fit at least the model to our question. No model is going to have it all. I will say that I will guarantee that. I'd be shocked if we ever come up with a animal model that simply has it all. So that's a very important question.
Very
All
good.
right.
Thank
I
you.
second that. I second that. I just want
Final
to
question,
paraphrase it.
Dr. Tagmeyer.
Statistical.
One more thought, David. I think one pervasive thought that you brought out in your lecture so beautifully is that the heart selects the most efficient fuels for a given environment. And this is adaptation. And then once this adaptation breaks down, becomes maladaptation, the consequences appear to be endless in general and very broad general terms.
Yeah.
Absolutely. I agree.
This is the one organ that not only needs more ATP than any other, it has to do it your entire life, and it has to be able to keep doing it. And the only way it's evolved to be able to do that, I think, is to be metabolically, multidextrous. And you keep chopping off various roots that, and yeah, it's a real driver.
And the chase, as far as I can see, will not end that soon.
No. Yeah.
Well, talking about fuel utilization, I think David, you just provide a lot of fuels to our continuing, quest for the complexity of the disease mechanism as well as potential therapies. And thank you so much. And I probably leave this to Dr. Maria to have the final concluding remarks.
Thank you. Gabriel, did you have a question? Did I interrupt you? I'm sorry. You okay?
No, it's fine. Please, we are already able to.
Yeah, all right, wonderful. David, thank you so much for fabulous. And it's wonderful to see you are so excited and passionate about this. Wonderful, beautiful presentation.
Yeah.
Grateful to Dr. Tagmeyer and Dr. Gabrielle Chiazzarella and Professor Yibin Wang, as usual, an old friend from Baylor days. Grateful to you all. Dr. Dr. Gerha, who is my next door. And again, I'm grateful to you all. It's a fantastic presentation. Grateful to you all. Thank you, David.
Thank you. Thank you,
Bye-bye.
guys.
Thank you all. Bye-bye.
Best wishes. Bye-bye.
Bye, everyone. Thank you.